Hello and welcome to my video on muscle physiology. So to begin, let's start with the sarcomere. So skeletal muscle at the fundamental level is made up of myofilaments, which make up myofibrils, which make up the contractile unit of a muscle, which is a sarcomere. Sarcomere means muscle part. So right here I've drawn the sliding filament model, which is basically, this is a sarcomere. This is on the borders, you have Z-disc proteins that serve as the scaffolding for actin and myosin, which are the contractile filaments. So there are myofilaments that are going to, at the molecular level, cause our muscle contraction by sliding over each other and shortening. So some of the molecular characteristics of the sarcomere. So let's start off here with troponin. So actually I'm going to start with F-actin and G-actin. So G-actin is the monomer that we're concerned with here. It has a binding site for ATP, and then when it hydrolyzes, it connects to form F-actin, and then that binding spot for ATP turns into a binding spot for your myosin head. So once these G-actin monomers polymerize and they form F-actin, filamentous actin, it forms a kind of double helix structure, which is then covered by a protein called tropomyosin. So now tropomyosin is going to inhibit the binding of these myosin heads unless we can get it off somehow. So tropomyosin is regulated by troponin. And troponin is a protein complex that consists of three parts: troponin C, troponin I, and troponin T. Troponin I has an affinity for actin, troponin T has an affinity for tropomyosin, and troponin C has 4 calcium binding sites, 2 high affinity and 2 low affinity. So what happens is when the cell receives when calcium is diffused onto our sarcomere here, naturally the 2 high affinity binding sites are bound because they're high affinity, and when there's low calcium, those high affinity sites are bound. And then when calcium diffuses from our sarcoplasmic reticulum down onto our sarcomere, those low affinity binding sites are going to be bound by that influx of calcium. And now we have all 4 binding sites on our troponin C that are bound. And what that's going to do is move tropomyosin off of our F-actin so that those myosin heads can bind and then do the cross-bridge cycle and the power stroke, which actually shortens your muscle and contracts it at the molecular level. But we'll get into that later. So we went over actin, tropomyosin, troponin, the regulatory structures, kind of the scaffolding. You've got your nebulin and your titin, you've got your Z-discs. And now we'll get into myosin. So myosin makes up the thick filament. Actin here makes up the thin filament. So myosin is a protein, and it has 2 heavy chains and 4 light chains. And these are little tails that wind around each other. And a myosin thick filament typically consists of about 300 of these myosin type 2 proteins. And all these heavy chains are going to interact, and they're going to create that thick filament. And then you have your hinge region, which is made up of your 4 light chains. And then you've got your head that's going to stick up, that's going to bind to the actin filament. Now the head of myosin has ATPase activity. So that means that it can cleave ATP into ADP and an inorganic phosphate. And then it has a binding site for actin there. And this is kind of what it looks like when it's bound. So you've got your filamentous actin with your tropomyosin and troponin, and then your myosin is going to come up and bind to that filamentous actin, undergo the cross-bridge cycle, and shorten. And that contracts your muscle. So now we have the neuromuscular junction, and this is how the influx of calcium is regulated to go into the cell, which is going to cause everything that we just talked about. So tying it all together here from last week, here we have our neuron, our axon, axon terminal, synaptic cleft, So we've got that voltage-gated sodium channel that propagates the action potential down the axon. And the whole point of that is to open this calcium channel right here. This is a voltage-gated calcium channel. Once the action potential reaches it, it is going to open and allow calcium to flow into the axon terminal. And calcium stimulates the exocytosis of these vesicles that contain acetylcholine. And it does this by interacting with the snare protein that is found in our neurons. So this little indented region here is on our muscle, on skeletal muscle, and this is our motor end plate. And our motor end plate is the region where we have nicotinic 1 receptors that receive acetylcholine, and this is important for our muscular contractions. So what happens is these vesicles of acetylcholine come out and they bind to these ligand-gated ion channels, these nicotinic 1 receptors that are going to allow sodium to flow into the cell and propagate our action potential. So those N1 receptors are on the border of what we call the sarcolemma. And this is kind of, the way I like to think of this is at a cellular level here. This is our muscle fiber, which is interchangeable with our muscle cell. So right here we have our sarcolemma, which we can think of as a plasma membrane. And then right here, this terminal cisternae on the triad, we can think of that as our smooth endoplasmic reticulum, but in terms of muscle, it's our sarcoplasmic reticulum, because we like to throw sarc in front of everything that has to do with muscle. But anyways, so that acetylcholine binds to these N1 receptors, propagates our action potential, and then that action potential is going to spread across the sarcolemma and then down into what we have here, which are these little indentations called T-tubules. The little indentations into the sarcolemma, and they contain extracellular fluid, and they're bordered on each side by a dilated region of the sarcoplasmic reticulum that is known as a terminal cisternae. And there's 2 of these, and together the 3 of these structures, our terminal cisternae and our T-tubule, make up the triad. So when that action potential spreads to the T-tubule, remember, this is our cell membrane right here, so it's voltage sensitive. And so there are these little DHP receptors, they are calcium voltage sensitive proteins that are going to interact with another receptor that is on the sarcoplasmic reticulum that is called the RYR. So together, this is the DHP-R complex. So that change in voltage stimulates the DHP channel, which opens up the RYR channel of the sarcoplasmic reticulum, which is going to cause calcium to diffuse down onto our sarcomere. So there's a protein called calsequestrin, which is right there at the gate of that RYR channel, and it has a low affinity for calcium, and so it's going to bind that calcium right there at the opening of that channel, so that when this opens, the calcium can diffuse quickly down onto the sarcomere, because otherwise that calcium would diffuse throughout the sarcoplasmic reticulum, and our muscle contractions wouldn't be as fast. So yeah, then that calcium diffuses down onto our sarcomere, and then it's going to interact with our troponin that we just talked about, which is going to take off the tropomyosin and expose the myosin head binding sites on our filamentous actin, and then our cross-bridge cycle can happen, which is how our muscles contract. And then in order to stop the contraction here, we have this circopump, which is responsible for relaxation. So this is a pump, it's primary active transport, and it uses a lot of ATP. This circopump here is responsible for the most ATP use that our muscles use. So this is responsible for relaxation. So it's going to pump calcium back into the sarcoplasmic reticulum. And the reason that contraction is faster than relaxation is because it takes less time for that calcium to diffuse onto the sarcomere than it takes for the circopump to pump the calcium back into the sarcoplasmic reticulum. So in other words, diffusion is faster than primary active transport. And this is a little close-up of our triad. See, we have the T tubules, terminal cisternae. I drew the voltage-gated Na channels that propagate the action potential, and then the DHPR complex that opens up and drops calcium down into the cytoplasm, which diffuses onto the sarcomere. And then we also have an enzyme called acetylcholinesterase up here in the synaptic cleft that is going to terminate our signal, right? It's going to eat up our acetylcholine so that our muscles don't just contract forever. And then it's going to split it into acetate and choline. Acetate just kind of diffuses off into who knows where, and then choline is recycled back into the neuron. Choline is vitamin B4, and we use it to make acetylcholine, so it's important, and we need to recycle it, and we also need to eat it in our diet. So then we get the cross-bridge cycle. So this is what happens when you put these two things together. You put the introduction of calcium and the neuromuscular junction and all the molecular characteristics together, and you get this cross-bridge cycle. So this top line here is going to represent our filamentous actin, tropomyosin, troponin, all that stuff in just one little line. And then this little stick figure here is our myosin head, and that's our thick filament. So thick filament and then thin filament. So our resting state is our myosin head is in a high energy state. It has ADP at the top there on the head and a phosphate attached to it. And this induces a conformational change, which is going to have our myosin head in the cocked position. And then we go through all this process just to release this calcium here. This is the calcium diffusing down onto the sarcomere. And then that is going to take the troponin off the tropomyosin and allow our myosin head to bind to that actin finally. And so what do we do when we make bonds? Well, we release energy, so some of that is released in the form of heat. And then we break that ADP and phosphate bond, and then we get our power stroke here. So the breaking of those bonds, the phosphate and ADP leaving, causes a conformational change to the low energy state of the myosin head, which is going to shorten our muscle. And this is responsible for our contraction. So right here is where we get our movement. And then in order to, because we don't just want to bind once, right? Those myosin heads have to keep moving, keep shortening, keep binding. So we have to add ATP in here to get that myosin head to dissociate from the actin. And then what happens is, since the myosin head has a ATPase activity, it's going to take that ATP and it's going to put it back into phosphate and ADP, and then we get back to our resting state. And as long as we have calcium, then the cycle can continue. But once that circuit pump pumps the calcium back up into the sarcoplasmic reticulum, then we're not going to be contracting like this anymore. All right, so there does it for part one there. So let me go back to my laptop and just make sure that I covered everything there. Yeah. So the molecular mechanism of muscle contraction is that those myosin heads are going to grip the actin, change their low energy conformational state, which is to move back, and it's going to shorten our sarcomeres, which produces force and muscle contraction. All right, so discuss the various sources of energy utilized for muscle contraction. Okay. So our energy stores that we use for muscle contraction, so we're going to be using ATP. So we have cytoplasmic stores of this. We don't have a whole lot of this. We have enough for about 1 to 5 seconds of contraction. And then an advantage of this, though, is that we can use it for our eyes and vision because our eyes make fast little movements. and we don't have to use oxygen here. And then for creatine phosphate, so creatine phosphate can readily donate its phosphate to that myosin, and it takes it can extend the contraction about 10 seconds. It's a rapid phosphate transfer, and it doesn't need oxygen. And those creatine phosphates, so they require enzymes, and this happens at the M line of the sarcomere, which is right down the middle of our sarcomere. And then the other energy source that we have for muscle contraction is glycolysis. So this is the splitting of glucose. Our muscles our skeletal muscle stores glycogen in it for ready use, or type 2 muscle fibers. So this is going to be used for our type 2 muscle fiber fuel, and this is going to be fast twitch and explosive. We can produce a lot of force with this, but it only lasts for about 60 seconds here. And then for aerobic respiration, this is where we're using the mitochondria processes. We're using oxygen to make ATP, and this will last us hours for medium activity. And this is good for endurance and marathons and that sort of thing. All right, let me make sure that I covered everything. So with our aerobic respiration, we have a lot of energy available to the cell there. Glycolysis, not really. Creatine phosphate, not a whole lot. And cytoplasmic stores, we don't really have a whole lot either. Okay. So now let's talk about the two main types of smooth muscle. So for smooth muscle, we're going to have our single unit and our multi-unit smooth muscle. Single unit is going to contract as a unit. It consists of cells that are connected by gap junctions. And single unit is going to produce waves. Single unit is what we find in peristalsis. And this is going to be found in our gastrointestinal tract. So we move food from our esophagus through our stomach, intestines, and then out the other end. And then it uses pacemaker potentials, so they are independent of, or they can be independent of the nervous system. And then the other type is we've got our multi-unit. And this is going to depend on nervous or endocrine input here. Alrighty, so now let's go over the smooth muscle contraction. So in our phasic contraction, this is similar to smooth muscle contraction. So where we start, so the difference between smooth muscle and skeletal muscle is that you don't have troponin to worry about. So we don't need calcium to move troponin out of the way or we don't need calcium to move tropomyosin out of the way via troponin. So just by adding another phosphate to this myosin, we can push tropomyosin out of the way and then undergo our contraction. So it all starts with the introduction of calcium. And in smooth muscle, this can happen in a variety of different ways. And one of those ways is through a G protein coupled receptor, which is going to send a cell signal to the sarcoplasmic reticulum, which is going to release calcium from there. So that's one example of a way that we can introduce calcium into the cell. And so we have these proteins here called calmodulin. And calmodulin has 4 binding sites for calcium. And once those get bound, calmodulin is going to undergo a conformational shift into its active form. And then we have an enzyme called myosin light chain kinase. And what a kinases do, they phosphorylate. So what happens is this is usually inhibited by kind of a foot and mouth mechanism. But then once we bind our active calmodulin that is responsive to calcium, once we bind calmodulin in its active form to myosin light chain kinase, then myosin light chain kinase becomes active. So we start up here in our resting position, where we have our ADP and phosphate. And then what we're going to do is we're going to activate myosin light chain kinase, which is going to add an extra phosphate on there, onto our myosin. And then that is going to allow the myosin to push the tropomyosin out of the way, so we're causing a conformational shift. And then that myosin is going to bond to the actin. And then because of that bond, we lose our phosphate and we lose our ADP. And then we undergo our power stroke. The muscle contracts, the sliding filament model shortens. But we still have our phosphate on there, which is very interesting. And this is going to allow us to keep contracting here. So if we add this ATP to get it, because in order to keep contracting, right, we have to add ATP to get the myosin to unbind from the actin. So we add that ATP, and then the myosin comes off, and then we still have this phosphate right here though. And so if the phosphate stays on the light chain, we're just going to repeat the cycle because of this ATPase activity of the myosin head. It's going to hydrolyze that ATP and turn it into ADP, and then we're going to have two phosphates on there. And because this phosphate stays on, we are able to just come and connect right back onto the actin. So if the phosphate stays on after our power stroke and ATP, we're just going to come right back over to the actin there. But then we have something called Rho kinase. And Rho kinase is an enzyme that is going to phosphorylate our myosin light chain phosphatase and inactivate it. And so myosin light chain phosphatase is going to take this phosphate off. And if that happens, then we go into relaxation and we just have our ADP and phosphate until our myosin light chain kinase can phosphorylate it again, and we add the ADP, or we add the myosin onto the actin again. So what happens though is we have this graph right here. This is very important. We have the myosin light chain kinase to myosin light chain phosphatase ratio. So we see that as we have an increase of activity of calcium, we have an increase of myosin light chain kinase, because calcium is responsible for activating the myosin light chain kinase. But then we have a slow increase of myosin light chain phosphatase. And due to a number of factors, this can be sustained calcium influx, chemical signals, depolarization events, we're going to have an increase in myosin light chain phosphatase. So through kind of poorly understood mechanisms, through sustained calcium influx, we're going to cause myosin light chain phosphatase to activate. And this is going to induce a tonic contraction. So let me explain this. So this is our latch state, or a tonic contraction. So what happens here is as the activity of the myosin light chain phosphatase increases due to our sustained calcium influx, myosin light chain phosphatase is going to be able to intervene earlier in the cycle here. So we have our whole myosin light chains kinase cycle causes ADP to bind there, but then what can happen is myosin light chain phosphatase comes in, and after we bind, right, we lose the ADP and the phosphate. Well, myosin light chain phosphatase is going to take that phosphatase off early, and then we just have our myosin head that is bound to actin with no phosphate on there. And so then what happens is if we add ATP to this, because of the conformational change, it's a lot harder for ATP to bind to that myosin head to get it to detach from the actin. And so it takes a while. It still happens, but during that time you have your relaxation, which is very slow, which is indicated by that wavy line. And then eventually, once that ATP binds, then the myosin head is going to do its ATPase activity, and we're going to get our ADP and phosphate again. But that's our latch state, is when, due to that conformational change, the ATP cannot bind and take the myosin off the actin very well. Alrighty, so remember, sustained calcium influx increases myosin light chain phosphatase activity, which induces a tonic contraction. So a tonic contraction is useful for our sphincters, right? Imagine if your sphincter, because tonic contractions don't really get tired. They can stay contracted for a long time because that myosin head is not leaving the actin. It's keeping that sarcomere short because it's not leaving. And so that allows it to basically never be able to get fatigued. So then our comparison of smooth and skeletal muscle here. So, one more thing. So our phasic contraction is good for our GI system, right? We don't want that to stay in a latch state. We don't want that to contract forever because then we'd get really nauseous and probably just vomit up everything that we ate. We just want a nice, smooth contraction to be able to move that food down our GI tract or to be able to close our blood vessels or our bronchioles. We don't want those things to become tonic. We don't want that to become a tonic contraction to where it stays contracted and constricted. So tonic is useful for our sphincters. So now, comparison between smooth and skeletal muscle. So with smooth muscle, it is unstriated. It's built a little differently from skeletal muscle, which is striated. And then skeletal muscle has troponin. Smooth muscle does not. Smooth muscle is found in sphincters, organs, blood vessels, the eyes. And then skeletal muscle, right, is found on the muscles of our skeleton. And in common, they both have sarcomeres. They both use actin and myosin. And so there's just a couple little differences there between the two of them. All right, so let me go back to my laptop and make sure that I covered everything here. Let's see. Okay, so finally we have the muscle sensory receptors. So we have our muscle spindles, and then we have our Golgi tendons. So right here, this big muscle we can think of this as our bicep, and those are made up of extrafusal fibers innervated by alpha motor neurons. And then we have our intrafusal fibers, which make up our muscle spindle, which are innervated by gamma neurons. So if we zoom in on our muscle spindle, we see that we have a couple types of fibers there. We've got our chain fiber and then our bag fiber. And it's called a bag because the nuclei are condensed in the middle and they make it bulge out like a bag. And then we have our chain, where it's a little more similar to a normal muscle fiber where those nuclei are kind of shoved to the outside. So then the bag receptors they're dynamic. So they are sensitive to the stretch of our muscle. Actually, like if I stretch my bicep and flex my bicep, the bag is going to be sensitive to that. And then the chain is static, so it's sensitive to those isometric contractions. And we're using 2A and 1A sensory fibers here. And then these muscle spindles have something called alpha-gamma innervation. So this is because when we stretch out that muscle fiber, or the muscle, we don't want our muscle spindles to go limp. And so we keep them innervated so that they don't just go limp and they can stay tight and continue to respond to the stretch of our muscle. And so then the way that this is incorporated is with the stretch reflex. So in the stretch reflex, say we have a quad and a hamstring, and if you go to the doctor and they hit you on the patellar tendon, what happens is that stretch in your tendon is sensed by this muscle spindle, which has a sensory neuron in it. That sensory neuron synapses on the dorsal horn of the spinal cord, and a signal is going to go up to the brain that tells you what happened. And then we're also going to synapse on some interneurons here. And one of those interneurons is going to be inhibitory, which is going to inhibit, it's going to relax your hamstring. And then the other one is going to contract your quad. And all of this happens due to interneurons because it is not under voluntary control. So you get tapped on the knee, and then a sensory neuron goes to your spinal cord, which synapses on an inhibitory neuron, which is going to rest your hamstring, and then the other neuron is going to contract your quad. So that's your stretch reflex. And then you have a Golgi tendon reflex. The Golgi tendon reflex is used to protect yourself, right? So if you didn't have this Golgi tendon reflex, if I were to flex my quad, basically what would happen is I would rip my quad off of the bone, I'd break bone. So we need to have a reflex here to get us to chill a little bit. So we only ever, so basically most of the time we're only acting, with a lower capacity of our true force that we can produce with our muscle so that we don't hurt ourselves. And then if you ever see if you ever hear a story of a mother deadlifting a car off of her children, her central nervous system is overriding these Golgi tendon reflexes and allowing her muscles to produce the most force that they can. And oftentimes that results in severe musculoskeletal damage if we produce the most force that we can. So we have this Golgi tendon that's going to regulate our force production. So what happens is we have a sensory neuron in our quad. It senses we are contracting. It's going to synapse on the posterior horn of our spinal cord, and we're going to interact with a couple of interneurons here. One of these interneurons is going to be inhibitory, and it's going to tell our quad to chill so we don't break our femur. And then the other one is going to activate our hamstring, because the hamstring is the antagonist to the quad. So that's our Golgi tendon reflex there. And those muscle spindles lie mainly in the tendon on the Golgi tendon reflex. And then we have our withdrawal reflex. So this happens when, say you're about to step on a sharp object like a nail, and maybe even you do. So what happens is you have this stimulus, this pain stimulus, and that sensory neuron is going to go from your foot. It's going to synapse in the dorsal horn of the spinal cord, and we're going to interact with a bunch of interneurons here. So on the leg that you're stepping on the stimulus with, we're going to synapse. We have an interneuron that activates your flexors. So it's going to lift your leg up, and then it inhibits your extensors. So it inhibits you from further pushing down on that sharp object. And then on the other side of the spinal cord, for your other leg, so if we were to just impact the leg that's stepping on the stimulus, we would probably fall on our butt. But we need to keep our balance, so we also need to have an effect on the other leg that keeps our balance. So we're going to use interneurons to do that. So our original sensory neuron is going to go to the other side of the spinal cord, which is responsible for the other limb. And we're going to synapse on an interneuron that generates an inhibitory postsynaptic potential that is going to make the flexors of that other leg relax. And then we have another interneuron that's an excitatory postsynaptic potential that is going to cause our extensors on that balancing leg to contract. So it's opposites there on each side. And this is used for avoiding tissue damage. All right, so let me make sure that I covered everything, and I think that should be it. I think I finished that up. Okay. Awesome. Well, thanks for watching my video. Have a good one.